So since I'm not recording this section, when I start lecture, remind me to record, okay? What is significance of the factor DNA A won't bind or C unless both DNA strands are methylated? The answer is to prevent reinitiation of DNA replication before one full run of DNA replication has been completed. The orders of the questions, the orders of the options are all different for different versions, but the questions are the same. How do beta rings facilitate DNA replication in E. coli? The answer, there are two answers, so it's D and C. They increase the rate of DNA synthesis, they increase the processivity of DNA polymerase total enzyme. Number 3, both prokaryotic eukaryotic ligases function by sequentially transferring which of the following chemical groups from cofactor donor to ligase and then to the phosphate of the next DNA. So it's A and P. Number 4, which of the following accurately describes both DNA A in the shape melting at or C region of the E. coli DNA? It is this one, DNA A binds and then DNA at or C, leading to loss of negative supercoil in the region. How do magnesium ions help catalyze the DNA polymerase reaction? Is again 2. They promote nucleophilic attack of 3' end of the last incorporated nucleotide on the alpha phosphate of the incoming nucleotide, as well as they facilitate the dissociation of pyrophosphate after formation of phosphodiester bond. This is tricky. The image was a little fuzzy, but it doesn't really matter. All you need to see is which component of anti-clockwise replication fork is trapped when it encounters termination site, indicated by arrow in the figure below. So anti-clockwise is over here, and so this indicated arrow shows the clockwise fork trap. So the answer is E, none. It will not be trapped since the fork will go through this termination site. In the E. coli mismatch repair process, which protein or proteins responsible for recruiting to the case2 DNA region being repaired is MUT-L. Which of the following statement about 4 enzyme components at replication fork is incomplete? Everything else is correct except the D, the DNA G, the polymerase, loaded onto topoisomerase and they travel together at the replication fork. The G is distributive enzyme, it goes on and off, so D is incorrect. And so this question has 3 different versions, and over here, you just look over here for now. You use the plasmid shown below in an in vitro DNA repair assay. You incubate the plasmid with extracts from normal human cells for 30 minutes. After repair, digest the plasmid with which enzyme would yield 2 DNA bands in your assay. Basically, over here you have BAMH1, it will always cut. The second is which enzyme allows you to cut this after mismatch repair, to cut this site. So if you want, because the human eukaryotic, you use mix-directed repair, so it repairs the strand, the outer strand, and it repairs C to A. So this restores the cell 1. So over here, to generate 2 DNA bands, you need the cell 1 and the BAMH1, because it will not repair ECAR1, it will repair cell 1. So the correct answer will be cell 1 and BAMH1. Version B, similarly, it will repair CLAR1 and BAMH1. The version C, CLAR1 and XCOL1. It's the same principle. Which of the following eukaryotic DNA polymerase incorporates both DNTPs and NTPs during DNA synthesis? And it's DNA polymerase alpha, which serves as a primase, as the primer, RNA primer, as well as the DNA primer. Which of the following statement about eukaryotic DNA replication is incorrect? It is C, eukaryotic DNA replication removes RNA primer by RNase H1, which is the same for eukaryotic DNA replication, is wrong because it requires FAN1 in eukaryotic DNA replication. 12. Terminal restriction fragment TRF assay utilize which of the following experimental methods to measure average telomere length in cultured cells? It's genomic sorghum blot. And this one, after cutting with restriction enzyme, you need to use genomic sorghum blot to detect the bands. Which of the following does not have a role in protecting telomere ends? It's the RB. On the graph below, depicting the shortening of the telomere of the cell culture, at which point you find the most senescent cells is at the Hayflick B, so the answer is B. I didn't highlight, but it's B. In a lab setting, which is or are minimal experimental steps you would take to immortalize somatic cells in culture? You have to express a TERT or human TERT gene in those cells because somatic cells does not express a protein component of polymerase, which is coded by TERT. Below are restriction sequences of 4 restriction enzymes, and identify all the enzymes that generate compatible ends that can be ligated together. For version A, it will be EAG1 and PSPOM1 because it generates compatible ends. For version B, it will be PST1 as well as ACI1, this one. And for version C, again, it's the, let me see. No, this is wrong. This is wrong. You can already see. Okay, which one? It's a GGCC. So it's this one. It's not the final version. I'm just giving you examples. So this, let me see if I can color it. So it's the EAG1 and the PSPOM1. Okay. So based on the restriction enzyme consensus sequence showing the question, which enzyme would you use if you wanted to create one end from the following restriction site? After cut by PSPOM1, it will generate, generate 5' overhand? If it generates 5' overhand, you need the T4 DNA polymerase to extend the 3' end to make one end. So the answer is A. For version B, APA1, it cuts over here between the 2 C's. What it generates is a 3' overhand. So you need to use a 3 to 5' exonuclease to true back. In the notes, it says a cladno fragment, but it's a 3 to 5' exonuclease. Version C, EAG1, it cuts again 5' overhand. It needs a T4 DNA polymerase. In part sequencing, which enzyme is used to remove the remaining DNTPs from the previous run before additional next DNTPs is a pyrase. Which reagent or experimental approach common among all 3 DNA sequencing techniques discussed in class, including Sanger, power sequencing, reverse terminator sequence? Which one? It's a synthesis DNA strand complementary to the template. It's the DNA synthesis-based assay, all of them. So if you're using the following pair of primers for a PCR reaction, which of the annealing temperature would you choose to use ensure proper base pair with template DNA, minimize unspecific binding. So for the version A, you all use the same equation, 2 times number of AT plus 4 times number of CG, and you have to calculate for both of the primers. Then you choose the lower melting temperature of the primer. Then you have to adjust the temperature to 3 to 5 temperature lower. So over here, 64, 58 is 58. Then you have to adjust a little bit lower, is 54. For version B, it's a 64, 66. Then you adjust lower, it's a 61. And for the version C, it's a 68, 66, then the lower is a 61, okay? So these are the correct answers. 21, for hybridization-based methods, such as microarray, which are following wash conditions will be optimal if you want high stringency in binding the DNA samples to microarray oligonucleotides. The wash condition, stringent wash condition will be higher temperature and low salt concentration. Which reagent commonly used to specifically pull out messenger RNA from total RNA in cell extract? It's oligo-DT linked beads. You mistakenly set up a PCR reaction using E. coli DNA polymerase 1 instead of Taq polymerase. Which step of PCR process would the reaction fail? It's extension. Because after heating, then the enzyme is nature. The final question, which of the sequence of newly synthesized DNA based on power sequencing results shown below? And for version A, it's GCTGGAA or GGCCA. For version B, it will be ATCAATTG. And for version C, it will be CAGCCAAT. Okay? For short answers, the order may not be the same for all different versions, but the first, the question regards to Grider-Blackburn in vitro telomerase assay. Why did the Grider-Blackburn select Tetrahydrina as a model organism? Because Tetrahydrina has a large amount of telomerase in their cells. Why did not Grider-Blackburn use the somatic cells for this type of experiment? That's because human somatic cells does not express functional telomerase activity, so it's not a good source for purification of telomerase. As long as you say that it does not express functional telomerase, you get the idea. See, in the in vitro assay, they're able to detect a ladder of radioactive DNA bands that indicate the extension of the telomere primer in the assay. What does ladder pattern indicate in terms of mechanism of the synthesis of the telomerase, telomere by telomerase 2? So either you have to say this is a repeated cycle of runs of a synthesis or stepwise pattern. Second, it is synthesized in increment of 6 nucleotide, or each run you add a hexamer or 6 nucleotide. Explain the function of a TERT and TERT. So the TERT is a protein component that is, as long as you say protein component that's synthesized or that catalyzes the reaction, and the TERT is an RNA component that used as a template for the synthesis. The second question is the Taqman qPCR. So you are using a Taqman PCR to monitor 2 different Omicron variants of the SARS-CoV-2. Then you collect the RNA samples from 50 patients, reverse transcribe RNA genome to double-stranded DNA, combine sample together, use a combination of a viral forward reverse primer along with variant-specific Pacman probe. Actually, over here, very specific already give you a clue how it works. For real-time PCR analysis, first list the structural feature or component of a typical Pacman probe. So you need to show that it has a reporter and a quencher that works together to give the fluorescent-like signal indicate the ongoing PCR reaction. You also have to have oligo regions that have a sequence complementary to the target gene. So describe the mechanism, Pacman-based qPCR mechanism, and in your response, explain how structural feature of Pacman probe allows detection after PCR amplification. So you have to include this 4 steps because it has a reporter punched together and it's not fluorescent. And the Pacman probe, it has a region that complementary to the template, so it will anneal to the template. Then when the ongoing active PCR, the DNA Taq polymerase encounters the Pacman, its exonuclease will actively chew or degrade the Pacman probe. This will release the R from Q, and that allows the R to fluoresce. And the next, we will have 3 different diagrams for the PCR plot. And so you use these 2 Pacman probe to detect the 2 different Omicron variants. EG5 or BA1 in the same reaction. What are the critical features of these 2 Pacman probe? And what is the name given to this type of PCR? So the key to detect the 2 different reaction, 2 different variants in the same reaction, you need to have Pacman probe, 2 Pacman probe. Each of the probe will have a sequence specific for one of the variant. Second, you have to use a different fluorescent reporters or 2 different colors. And this type is called multiplexing or multiplex PCR. You perform a qPCR, the results are shown in the figure below, which is above, and you can see whatever. So for A, the EG5 is more prevalent, more abundant because it has a lower CT value. For the B, EG5 again is more abundant. In C, BA1 is more abundant. And how many more fold of dominant variant is present in the sample than the less dominant? So you have to write out the correct CT values and then calculate using equation 2 to the power of delta CT. So the fold is 128. Last one is the Sanger sequencing. So you use the Sanger sequencing to sequence the DNA strand where you know the sequence of the first 5 nucleotides coming from the 3' end of the molecule. Use knowledge to do the design primer and use radioactive labeling and auto radiography to visualize sequencing gel shown below. So over here, this is A, this is B, this is C, asking you the lane 3. So in the sequencing reaction beside DNA template and primer, list all other reagents that you will need to mix and incubate together to produce products for lane 3 of the sequencing gel showing there. Be specific and include the relative amount. So you need to say the DNTPs or equal amount of all that. This has to be in excess or large quantity. You have to also say the small amount of DDNTPs or dideoxy, the specific nucleotide based on your image. Because for this one it will be DDTTP, for this one will be DDGTP, for this one will be DDTTP. And then you also need to include a little bit the radioactive form of these DNTPs and DNA polymerase. And then to derive from the image of the synthesized strand and the template strand, so the synthesized strand, you read from the bottom up, from 5 to 3 prime. So over here, it's a GTA, so on and so forth. And for the template strand, it's a reverse complementary as long as you label polarity, whether you write it from 5 to 3 or 3 to 5. As long as the sequence showing the complementary to the synthesized strand, it's all